Hello everyone. So today we are going to learn about different types of microbial plating techniques. Okay. So moving forward, microbial plating. What is microbial plating? So before diving into that, we need to know that microorganisms are present everywhere. Right? They're present in high numbers everywhere, whether it's air or water, soil, deep ocean vents, volcanoes, sand, or any living creature. Okay, they are present everywhere. So, in that is also in high numbers. So, this microbial plating, what it does, it helps us to grow these microorganisms, these invisible microorganisms in a media, and helps us to study their various characteristics. Okay. Now, these characteristics like morphology, colony counting, pure culture, isolation, shape and size of the microorganisms, texture, and their arrangements. Okay. So, this plating can be made for different types of microorganisms that may be bacteria, fungi, or viruses. Okay. So, this is the microbial techniques. Now, what is the importance of microbial technique? What it will do actually? Okay, so this technique will aid in growing the invisible microorganisms on the Petri plate. Okay, it will help us to identify the types of microorganism, to study it, and to isolate it according to our interest. It will allow the pure culture of the microbes of interest and grow their culture in vitro. Okay, so from here we can also do the pure culture of our interest. Okay, so microbial plating, it will also help in the microbial preservation. Suppose the species, this is the important species that you have discovered and you want to preserve it. Okay, so you can also do it. You can do the stock culture and you can submit your species for identification. So that is how. So this plating will help you to store the microbes of your research interest for a very long period of time. Can also reveal novel microorganisms, as I said, which are not discovered yet. You can also reveal novel microorganisms. Study of the pure culture using microbial plating method, it can decode the merits and demerits of the particular specimen. Right, we can study their nature, their virulent or non-virulent nature, everything, their gene structure, everything can be studied. So, coming to the types of microbial plating. What are the types of microbial plating that are used commonly in our microbiology lab and the scientists who have invented it? So there are three major microbial plating techniques. First one is the spread plate, then the streak plate, and then is the pore plate. Okay, and these methods was developed by two bacteriologists, Loeffler and Gaffkey, where in the laboratory of Robert Koch. Okay, so Loeffler and Gaffkey invented these major techniques to isolate the microorganisms. Now, in this video, we will learn only about spread plate and we'll learn the other plating methods in our next video. So, spread plate is a very basic microbiology technique which is used for isolating and counting the viable microorganisms that are present in a liquid sample or on a soil sample. If you are using a soil sample, you have to make a liquid suspension of it. Okay. How? By spreading a certain volume of a sample on an appropriate solidified culture media. Okay. By this you can do the spread plate. It will be clear in our next slides. Now here this method is suitable for growing aerobic and facultative aerobic microorganisms. It is an easy, simple and economical method. Now, what are the requirements? So, first requirement obviously is our culture media, which contains the agar. Second is our the sample of interest, whether it's a liquid sample or a solid sample. Next is our flask, 50 or 100, in case you need to do the mixing. You need a Bunsen burner, you need test tubes, micropipettes, petri plates, incubator, autoclave, distilled water, glass or plastic spreader, and 70% ethanol. So these are the requirements that you need for the spread plate technique. Now coming to the procedure. Now here in the step 1, you need to prepare the required agar media and autoclave it at 121 degrees Celsius 15 lbs for 15 to 20 minutes. In step 2, the petri plates are poured with agar media in laminar and allowed to cool for 20 to 30 minutes. In step 3, you need to prepare the sample of your interest. Okay, so if you are having a liquid sample, then it's good to go. But if you are taking any solid sample, then measure 1 gram and add it to the 10 ml of distilled water in the flask and mix it well. So if the soil suspension is very dense, you can also do the cereal dilution. Um, the cereal dilution has been covered in our previous video. The link is in the description. You can go and check it out. So in step 4, take 0.1 ml from the sample using a pipette and drop it on the agar plate. And then you spread your sample evenly on the agar plate using the spreader. You need to make sure that you do not push too hard on your agar plate so that the agar will get broken. Okay, so you need to do very softly over. You just need to spread the culture over your media till it gets completely absorbed and then label it after. Then keep the plate in the incubator in inverted position at given temperature for a specific time period. Generally 37 degrees Celsius and 24 to 48 hours it takes. Okay, so this 37 degrees Celsius is common for all types of microorganisms to grow, and they basically grow between 24 to 48 hours. Within 24 to 48 hours they grow. Okay, now observe the colonies next day or day after. Now this is the picture procedure. If you take the soil sample, you need to make a liquid suspension. Please remember that. This is a liquid sample. Now from here, you need to take 0.1 ml and put it in your agar plate. Then you do the spreading and then you put it in the incubator in the inverted position. Remember that also. And then you observe for the colonies after the time interval, that is 24 or 48 hours. Now here are some of the points that you need to remember is that you need to have open the lid of the petri plate. So okay, so you should not uncover the whole petri plate at once. Okay, it's better to hold the lid half open with your thumb and the index finger and do the spreading. Second is to hold the spreader at the 45-degree angle and you move it back and forth after touching the drop of the sample that you have put using the pipette. Okay, so you move it back and forth. Now rotate the plate with your other hand and you continue the back and forth movement to not miss out any corner. Now leave the plate for 5 minutes for it to get absorbed completely in the laminar only, and then you label it properly and then you put it in the incubator in an inverted position. Okay, so these are some of the things that you need to keep in mind for spread plate technique. So the spread plate technique is commonly used for morphology assessment, isolation of bacteria, and for pure culture. Now here first we will see for morphology assessment. We'll study morphology assessment first. Now let us understand using an example. Now suppose you have a sewage water sample and you want to see the different types of microorganism present based on their morphology. Now how will you find it? So as we have studied the procedure of the spread plate, the technique is same. You prepare the culture media with agar and then you plate the media and allow it to solidify it. After autoclaving, then take 0.1 ml from the sewage water and drop it on the media. Now you do this using a micropipette. Okay. Now spread the drop evenly over the media using a spreader until it goes completely gets absorbed, right? And then you put it in the incubator at 37 degrees Celsius for 24 to 48 hours. In the inverted position, of course, and observe the time taken for different types of microorganisms. Okay, now since you have taken the sewage water, their many types of microorganisms may be present, right? There may be bacteria, there may be fungus, there may be virus. Okay, so different types of microorganisms may be present, and all these different types of microorganisms might take different times interval to grow on the plate, right? So you need to observe the time that they're taking to grow on the plate. So what can we observe after the incubation period? Okay, so what is expected in the observation actually? So the plate which is shown in this picture, we can expect this type of microbial growth. Okay, so here in this plate we can see there are different types of microbial colonies have grown. Based on their morphological characters we can identify it and we can count it. So here the total plate count is done by counting the number of colonies based on their morphological characters. Okay. So here we can see that there are yellow colored round shaped colonies present, which are 5 in number. So we'll count different types of colonies based on their morphology. Now here the yellow colored are 5 in number. Similarly, off-white colored round shaped colonies we can count, they are 8 in numbers. Yellow colored rod shaped colonies are there, which are 3 in numbers. Blue colored small round shaped colonies are present, which are 12 in numbers. You can also count it from the plate. And besides this, there are also fungal colonies present, where off-white colored irregular shaped fungal colonies are present. They are 2 in numbers. And blue colored peach shaped fungal colonies are present, which are 4 in numbers. So the total plate count is the addition of all these types of colonies based on their morphological characteristics. Now if we add it, we will get a total of 34. So here we can say that the total plate count is 34. Okay, so this is how the spread plate is done from an unknown sample. Now coming to the isolation. So spread plate technique, as I said, is also used for isolation and pure culture. So coming to the isolation and pure culture method. So suppose now you have got the plate. Now from there you want to isolate one type of bacteria or fungus and you want to make a pure culture of it. So how to do it? Now the process is same. Now here you prepare the nutrient broth and nutrient agar. Here the extra media is prepared, the nutrient broth. So you prepare the nutrient broth and agar, you autoclave it. You do the solidification of the agar media in the plates and you cool down the broth media to the room temperature. Okay. Do not use the broth when it is in hot, because after autoclave the broth will get hot, the temperature will rise, so you need to cool it down to the room temperature before you use it. Now what you do, now you do the isolation. How? You collect the bacteria or the fungus of your interest from the plate that you have got from the spreading previously. So you collect the bacteria or fungus using an inoculating loop and you mix it to the broth media that you have prepared. Okay, now allow the growth of the bacteria in the incubator. You keep your conical or the flask that you have taken, the broth media which contains the broth media. Now we have put the bacteria or the fungus of your interest in it, and then you need to keep it in the incubator. You need to keep it in the shaker incubator. Okay, to enhance the aeration. Okay, but these are majorly they are aerobic bacteria, right? So they need aeration. So you put it in the shaker incubator. Now here as it is shown, you this is the plate that we have got. You, for using an inoculating loop, you take the colony of your interest and you put it in the broth that you have autoclaved. Okay. Now next you take 0.1 ml of microbial culture from that particular plate the next day and you spread it on the petri plate. As shown in this picture, you spread it on the petri plate and spread it till it gets completely absorbed. Okay. Now this will become your pure culture. Now if you find that the same types of colonies are growing on your plate, now this is the pure culture. Pure culture of that particular colony that you have taken from that previous main plate. Okay. Now you can make another broth culture out of it and you can do the storage. You can store it in the fridge as a stock pure culture. Okay, and you can also identify the bacteria later on using gene sequencing or other methods. Okay, so this is the basic method that we have used for the identification, that is the morphological assessment we have done. For deep identification, you need to do the gene sequencing, which you can do according to your need. Okay, now what is the observation that you can expect? This is the main sample plate. You have taken this colony of your interest for isolation of bacteria. Now you have isolated the microbe. Now you have done the pure culture. Same types of colonies you will get on the plate. This is the pure culture of the isolated microbe. And then you will do the stock pure culture of the identification. You can take this colony and you can do the broth again. And this will be your stock pure culture. Okay. Now, precautions that you need to keep in mind before doing this spread plate technique. First, you need to keep in mind that you need to heat sterilize all the glasswares before use. Okay, and if you are doing serial dilution, you need to do the sterilization of distilled water as well. You do the autoclave if you want to do the serial dilution of your sample. Okay, so you do the heat sterilization and you do the autoclave of your distilled water as well. Now use gloves and apron to avoid any spillage. Autoclave all the media, the petri plates, the broth, distilled water, everything that you will use. Always use 70% ethanol to sterilize your hand and work area in the laminar hood. Okay, the work area that you are working on, you need to clean it, and before that you need to sterilize your hand as well. And heat sterilize the inoculating loop and spreader using Bunsen burner. Allow cooling down before use. Do not use the inoculating loop when it is hot or spreader when it is hot. Okay, you cool it down and then you start your spreading or taking culture using the inoculating loop, right? The sixth one is always use calibrated micropipette. So you need to take 0.1 ml. So before that you need to calibrate your micropipette to 0.1 ml. Okay, otherwise you will not get a appropriate amount. So you need to do the calibration for drawing the culture. 7 is to keep the culture plates in inverted position in the incubator. In the incubator, you need to keep it inverted. Okay. And you need to clean the working area after completion with 70% ethanol. So clean your working area with 70% ethanol after you have finished all your work. Now, also this is the most important one, the ninth one. If you take a soil sample, please remember the sample should be mixed in a liquid or need to be made in a liquid suspension. Okay, so only liquid sample can be used for this spread plate technique. Okay, so that is all for today's video. If you have any queries regarding this, you can comment down below. If you want the detailed notes of this video, please follow our LinkedIn page. The link is in the description as well. And if you like this video, give it a thumbs up and do share and subscribe to our channel for more such related microbiology updates. Hope you have enjoyed this video. I'll see you in our next video.